Alright guys, we're going to go ahead and finish out section 10.1. And it's mostly going to be learning how to do Punnett squares and interpret probabilities in your crosses here. So these are called Punnett squares. And they are tools that we used to make predictions about what will likely happen, not necessarily what will happen. And we'll talk a little more about the difference between a prediction of what will likely happen and necessarily what will happen. And these are easier to learn by doing. I got 4 examples. Hopefully by the time we're all the way through they make sense. When we're talking about probability, it is how likely it is a certain event will occur. And we can use probability as a percent, like 25% likely to happen, or as a fraction. I could say there's a 1/4 chance, or as a decimal, I could say 0.25. Right? So when we're thinking about this and this and this all mean exactly the same thing when we're talking probability. Alright, so here's an example with a Punnett square. In P's, yellow is dominant over green for P color. So since yellow is dominant, we are going to use a capital Y for yellow because we always use the dominant, the capital letter of the dominant version of the allele. And so for green, we're going to represent not with a G, but with a lowercase y. And I cannot emphasize enough how important it is. A lot of letters, the only difference between a capital and a lowercase is the size. So you need to figure out some way to reliably tell your upper from lowercase letters apart. So you notice my Y, I may make a little curly Q, and I know if I see a little curly Q, then it's lowercase. I mean, think about some of these other letters, like S. You tell me, capital or lowercase, right? So you have to think about, okay, if I'm going to use the letter S, I'm going to make my capital S is really big and my lowercase s is really small, so I know which one's what here. But back to the example here. If a heterozygous plant is crossed with a plant that produces green peas, what are the possible genotypes and phenotypes of offspring, and how likely are they? Okay, so these are our parents here. So we want genotypes of the parents, which remember is the genes that they have. Okay, so we have one plant, one of our parents is heterozygous, which means it has 2 different alleles. So it might be capital Y, lowercase y for this one. That's the 2 different ones. It's crossed with a plant that produces green peas. So green is recessive. So the only possible genotype that would show the green recessive version is if there are no dominant alleles around. So it has to be lowercase y, lowercase y. That's the only thing that makes sense if it produces green peas. So phenotypes, this is me physically, what are they going to show? So this one has a dominant allele. If there is at least one dominant allele, that's the version of the trait that shows up. So this one would make yellow peas. And this one does not have any of the dominant allele, so it would make green peas. Alright, so to use a Punnett square, you're going to take the genetics of one parent and put them on the side. And it does not really matter which parent you use, but we're going to split it up. So I'm going to put the capital Y here and the lowercase y here. The other parent, same idea, but on the top. So I'm going to take lowercase y here, lowercase y here. Then we just fill in the boxes with what is on the outside of the box on the side and what is on the outside of the box above. So this box is going to have an uppercase y and a lowercase y. In cases like this where you have one big, one little, we always put the capital one first. So same idea. This one, the capital Y from over here and the lowercase y from up here. So in this box, I have a lowercase y over here, and then again I look way up at the top to see what my other one is. So I would have little y, and in this box, little y, little y. And sometimes my curly cues don't go all the way through. So we have a box here split into 4 pieces. We know that one box out of 4 would be what percentage? Right, 1 out of 4 would be 25%. So we're going to figure out in our offspring what are the possible genotypes and what are the possible phenotypes. So remember, genotypes is the genes. What possible gene combinations would be in this box? So I have this combination, big Y, little y. And I have this combination, little y, little y. Okay, in this Punnett square, those are the only possibilities. Remember, so out here are our parents. There's parent 1, parent 2. Each box is a possibility of their offspring. Okay, so what percentage would be capital Y, little y? Well, in 2 of the boxes out of 4, there's a big Y, little y, so that would be 50%. And in 2 of the boxes out of 4 are little y, so that would also be 50%. But when we're talking about phenotypes, we're talking what would physically present in each of the offspring. Okay, so we're not talking about Y's for phenotypes. We're talking about, are they making yellow peas or green peas, because that's what would physically show up. So we have yellow and green. So this one, so if the dominant version is yellow, any box that has at least 1, which, you know, that could be 1 or 2 capital Y's, would show the dominant version of the allele. So both of these have at least 1 capital Y. So again, 2 out of 4 is 50%, and these 2 do not have any capital Y's. So the recessive version would show up in, again, 2 out of 4, or 50% of those. So let's try second example. Ooh. So this time, smooth is dominant to constricted. So a smooth pea pod would look like this. Right, so there's my little smooth pea pod. A constricted pea pod, the pod kind of sucks around every individual pea. Okay, so they say it's constricted, it's pulled around. So smooth is dominant, so we're going to use capital S for smooth. And constricted is recessive, so we're going to use a little baby s for constricted. So remember, s's look exactly the same, capital versus lowercase, except for size. So exaggerate your size differences or you will regret it. Alright, so this time we have 2 heterozygous pea plants. Both our parents are heterozygous, which means, again, they have 2 different alleles. So they're each going to be big S, little s. Big S, little s. And I maybe exaggerated so much that I can't even tell it's an S. There we go. So that's the genotype, is the alleles, what the letters are that they have. And then the phenotype is the physical, what do they show. Okay, so they each have at least 1 dominant allele. So they would each show the dominant version of the trait, which is smooth. Alright, so again, we take one parent, take them to the side, big S, little s. Take the other parent, split them on the top, big S, little s. Again, I would encourage you to pause right here, fill those boxes in for yourself, and see if you agree with mine. And here's what mine look like. So this box should have big S from big S, big S. This one should have big S, little s from up here and over here. This one, big S, little s, big S over here, little s up here, and this one would be little s, little s. Okay, so there's your Punnett square. Remember, each box represents 25% probability. So now we're going to, again, try to figure out what is the probability of each of the genotypes in the offspring here. So I've got big S is one possibility. I also have some boxes with big S, little s. Then I have some box with little s, little s. Again, exaggerate the differences of your sizes of letters so you can make sure and tell the difference. So big S, how many boxes are there? Just one. So 1 out of 4 is 25% probability. Big S, little s, I have 1, 2 of those, so that would be 50% chance of that one. And here's my little s. So since there's no dominant alleles, then and only then, the recessive version can show up. So that's 25% of that one. Here's our phenotypes, our physical versions of the trait that could show up. So remember, for the dominant, the smooth, you need at least one dominant allele. So that could be 1 or 2. So if I'm looking for how many boxes have at least one capital S, I have 1, 2, 3 of them. So that would be 75%, and only one that does not, 25%. Alright, we've got 2 more of these. I'm going to go a little bit faster. Again, I would encourage you to pause right now and try this entire page 82 and then double check with me, but we're going to just kind of go here. So, this is the weird thing about this. Nobody's asking me questions. I'm going to go way too fast. So we're going to recreate the experiments done by Mendel. Okay, so remember we said he started with pure breeding parents. So pure breeding is going to work out to be the same as homozygous. Okay, so we're going to start with, we're going to do purple and white. So we had pure breeding. So we had one that was pure breeding purple, meaning it's only capital P for purple alleles, and one that's pure breeding white, meaning it has only lowercase p white alleles. So the phenotypes would be purple flowers for this parent and white flowers for this parent. Take the alleles of one parent and put them out on the side. Take the alleles of the other parent and put them on the top. And then we fill in, putting capitals first if it's going to have 2 that are different. Okay, so if you look, every single one of those boxes is exactly the same. Okay, so we have to think though, if I said, what is the genotype of the offspring? Genotype would be big P, little p, 100%. Phenotype would be purple flowers, again, 100% of the time. That's the only possible combination when we do that one. All right, so the next thing Mendel did was he took their offspring, these ones, and made them the parents of the next generation. So the genotypes of the parents of the next round are going to be the big P, little p. And since they have at least one dominant allele, we're going to say that they would have purple flowers because that is the dominant trait. Then we do the same thing here. So we're going to take one parent, split their alleles up and put them down the side. And our other parent, split their alleles up and put them on the top. And then fill in. Okay, and you'll get really good at these if you do that worksheet I sent. Alright, so genotypes. Again, this is the letters, this is the genes. So we have 3 combinations. We've got the big P, big P, big P, little p, and little p, little p. You notice my way that I tell my capital and lowercase p's apart is my lowercase t's have this little tail. So I've got one box with those, so that's 25%. 2 boxes like this, that would be 50%, and just one box like this, 25% again. But then when we're talking phenotypes, remember that's the physical trait that shows up, and I have 1, 2, 3 boxes that have at least one capital P dominant allele, so I would have purple purple flowering plants in 75% of the offspring, and white in only 25%. So all the Punnett squares that I did in this video have only one gene involved. So this one, the gene was for flower color. We did the gene of pod shape. We did the gene of pea color. But they all just have one trait, and so we call them mono hybrid crosses. Mono for one. They're just looking at one trait, and that's it for this video.